Hi, everyone. I'll be the host for this session and like to introduce Chibo. I will not try the surname. However, Chibo is currently the micro and the macroalgae section of SANOMARC. She has previously worked in various marine institutes in Namibia, such as Ministry of Fisheries and NSI in Walvis Bay. She has an MSc in marine microbiology from the University of Bremen and a PhD in marine biology from the University of Rostock. Chibo's interests are phytoplankton bloom dynamics in the northern Benguela Upwelling System, phycotoxin production in the NBUS, assessing the effect of ocean acidification on biological species, metal pollutants in macroalgae, and utilizing macroalgae for industrial purposes. So Chibo, the floor is yours. You can start with your presentation.
I'm going to be talking about how you can use multiple stresses in order to get an understanding of how your biological pump will potentially respond. And in this instance, I'm speaking specifically for phytoplankton. So I'm going to be using the following keywords interchangeably. So we'll be looking at the Benguela because this is the area that we're in. A lot of the work is going to be focused on local cultures that we're getting from here. The multiple stresses we'll be talking about, temperature is going to be one of them since it's related to climate change, macronutrients. Instead of writing ocean acidification, I chose to write carbonate chemistry because we're dealing with phytoplankton and photosynthesis, and then as well as phycotoxins because they will be affected by this, and then I will end off with dust. So because we're talking about the biological pump, one of the areas in which we sort of get its importance is by relating it to the carbon cycle. So, I mean, I showed this from my last presentation where we have anthropogenic carbon that is being outgassed at rates much higher than what is taken up by the marine environment as well as the terrestrial environment. But the area of interest here with respect to the biological pump is that you have an instance of where you will have the uptake of CO2 in the form of DIC in the water by the phytoplankton. That would produce particular organic matter, and that will be transported down into the deep ocean where it will actually be sunk. So from that perspective, you get an idea that the reason why the carbon cycle is so important is that this is a way in which we can actually try to mitigate the effects that we're seeing from anthropogenic carbon dioxide. Now, of course, because we're talking about primary production biological pump, we need to look at areas that have a high level of primary productivity, because those are the areas that would be most likely to be able to take up a lot of this excess carbon dioxide that we're getting from anthropogenic sources. So if we look at that specifically, we have the eastern boundary upwelling systems, of which there are 4. And these are the areas that have high primary productivity. So they're able to take up a large amount of the PCO2. And if you had to look at what these areas constitute of with respect to phytoplankton, they're actually dominated by diatoms, and they actually account for about 20% of this primary productivity. So again, as I mentioned before, this is where we're saying, fine, we have an area of high primary productivity, we're taking up the CO2, and we're able to transport it into the deep ocean where it can actually serve as a sink. Now, when we're talking about taking up organic anthropogenic carbon dioxide, we're actually looking at photosynthesis, where we're taking up DIC, so the dissolved CO2 that's the CO2 that's dissolved in the water column, that is taken up by the phytoplankton, and we form particulate organic carbon. So when I'm going to refer to the experiments that are going to be done with respect to this, I'm literally only speaking about this area of the biological pump. Now, because we're dealing with a biological system, even though it is very efficient with respect to looking at the vertical gradient of DIC to actually pull up from the atmosphere into the species themselves, it is very sensitive. and chances of destabilization are there, to the extent of where in some instances you'll actually have, instead of transporting the carbon into the deep ocean, you'll have it be remineralized and taken back up into the atmosphere. So instead of taking it up, we're basically giving it back into atmosphere again, adding to what is already there. So with respect to the biological pumps, they are of interest to us because they also aid in modulating the atmospheric CO2. And at times like this, when everyone's always talking about climate change, this is one of the factors that's very important in this area. So even though we know, yes, there's a biological pump, photosynthesizers, phytoplankton, we still don't really have an idea of the full understanding of, if we have increases in CO2, are our oceans just going to keep on taking it up because that's what they do when they photosynthesize. The understanding that we have of this aspect is very species specific, but there are a lot of areas that are still not known. So if we go from the pump itself and down to our area, the reason why the Benguela is so important with respect to this is that we tend to account for all the eastern boundary upwelling systems. We account for 40% of that primary production. So this is an area that has high biomasses of phytoplankton. And for those of you that were listening to the previous presentations, you know littorates. So you have the northern Benguela north of littorates and the southern Benguela south of littorates. And from data that we have at the moment, you have the northern Benguela as a carbon dioxide source, and the southern Benguela as a carbon dioxide sink. And the reason why the northern Benguela is a sink is because we have high rates of remineralization. So the particulate organic matter is broken down quite readily by the microbial community. And one thing to notice here is that, yes, fine, from this calculation, we are seen as a source of CO2, but the magnitude of difference between the southern Benguela and the north is marked. So if we had to look at the area, and these flux rates were determined using predominantly offshore samples. So it doesn't really factor in the variability that you would get from coastal areas. And with respect to the biological pump, those are the areas where you'd have actually the strongest, effects. And yeah, and that effect that we actually get, again, to reiterate from what I said before, is that it would be very dependent on the diversity that you'd get in that area. And Madagraph in 1978 sort of looked at a lot of these factors with respect to phytoplankton and realized there's some key factors / potential multi-stressors that you can put together that would actually help you predict whether or not you'd get a large biomass. And by large biomass, meaning that you'd take up a large amount of the DIC. And these key bloom conditions were light stratification of the water column, and if you had sufficient nutrient load. So these factors basically feed into what the phytoplankton would need in order to proliferate, basically. And an additional factor would also be looking at the extent of aerobic decomposition, in the sense that if that would be low, it would also enhance the movement of the DIC and allow the particular organic matter to actually be transported offshore. Now, as a Benguela, we tend to be very limited with respect to that the nitrate is actually the limiting factor when it comes to how efficient the biological pump can actually be. So I've been talking about blooms. I just wanted to take a step back to also just sort of categorize what I was actually talking about. So in this instance, we have about 300 species of the 5,000 known marine species. Only 300 of them actually produce HABs. And by HABs, we're classifying them according to either they produce a toxin, they produce high biomasses that deplete oxygen in the water column, or they actually have protrusions that can actually cause mechanical damage in species around the area. So if you look at this water, this is an example of a bloom that we tend to get quite readily here, where this was a dinoflagellate bloom. And if you took a Secchi disc down about like 20 centimeters, you couldn't see it after that. That's how thick the water was. So again, these are key, what do you call it, bloom drivers. The only addition here is the changes to nutrient availability, where that might actually key in specific species. So if we look at multi-stressors, so we've been talking about, temperature, PCO2, all these other aspects. Now we want to take a step further and say, fine, if we want to have an idea of how our biological pump will be affected by the environmental parameters, this would be the multi-stressors. So we're looking at the alteration of multiple environmental properties. Now these properties have to have a biologically influential effect that is linked to anthropogenic pressures. In that instance, that is what we would call a multi-stressor for this specific aspect. And of course, we would link it to climate change. So in that instance, you would look at, everyday stressors that we all know of, which is temperature, carbon dioxide, pH, oxygen. The idea of it is that we would be able to combine all of these aspects, because as we know, in your normal, your normal environment, you don't only have a change in temperature or, a change in oxygen concentration. You have a whole host of things that work together. And the advantage of using multi-stressors in this instance is that you'd be able to predict the potential impacts you would actually get from either increasing one, decreasing the other, and also looking to see whether they work synergistically or whether they work antagonistically with each other. And for, again, I keep on saying climate change, but these are the things that are of importance to the marine environment at this point in time, because climate change is something that we have to basically adhere to. Or not adhere to, we have to try to combat. I think that's a better term. Anyways. But again, even though we're looking at multi-stressors, we also need to be aware of the fact that we need to pick drivers / multi-stressors that are regionally distinct. Because if you take the northern Benguela, for instance, eutrophication would not be a multi-stressor for us, because we have our water has a high nutrient load when it's upwelled. And if you look at the coast itself, we don't really have a lot of terrestrial inputs into the ocean. So if we took the idea of multi-stressors and we want to take it one step down and look at laboratory and mesocosm experiments, this is primarily because if you look at the direction most places are going, they tend to be moving, not necessarily moving away from ocean-based sciences, but trying to sort of add other aspects to it, because getting space on a ship going all the way to get samples back and forth is not the easiest thing to do. And for example, if you wanted to track a bloom, because sometimes basically the bloom cycle is 2 days, it will cost a lot of money for you to stay in one spot and actually track that actual process itself. So this is where laboratory and idea of what is going on there, but what if we take it one step further, put it in a local environment and try to control certain aspects of it? This is where these experiments would come from. So we're trying to link between laboratory experiments as well as the basic mesocosm ones, where you'd actually be in the field itself, but you would have parameters in such a way that you'd be able to control certain aspects. Because that's the point of these sort of experiments for multi-stressors. You want to be able to say an increase in temperature causes this, a decrease in pH causes that. That's the whole idea behind it. Now, of course, the complexity of the mesocosm or the laboratory experiment that you would set up is dependent on what you have at your disposal. And it also depends on the kind of questions you were trying to look at. If you're looking at physical properties such as tidal currents, you would need something larger. Or biological complexities, you would need a variation of, let's say, multiple species. But again, depending on the questions you're trying to address, you would pick one of those aspects and add them according to that. So I just added two aspects to show you that you could go from really large This is the schematic representation where these are literally put into the ocean. You have a net that basically allows for the transport of water. So the water outside and inside are basically comparable. And here they're looking at the, what do you call it, species succession of the phytoplankton community and linking it to the herbivory with respect to the zooplankton. And then this is another Cosmos experiment where they had, I think it was off of Peru. So there also, again, they have those other aspects. So depending on what you are measuring, you would do the, what do you call it, the sampling and analysis, excuse me, with respect to that. And then you could have something as small as this, where this is basically 100 milliliters, and they're looking at how the variations in PCO2 is affected by my microbial community. So again, the extent with which you build your mesocosm / laboratory experiment is dependent on what you were trying to find out. So with this in mind, there's a series of 3 experiments that we're going to set up here. Now, a lot of what I'm going to say now would be best linked with the Walvis Bay. I'd say the Walvis Bay Harbor, literally because most of the work is related to the, what do you call them? The mariculture industry. So yes, these findings could be linked directly to a biological pump offshore, but it's the thought process is more for enclosed coastal areas, basically. So the idea of this first experiment would be to look at the effects of the multi-stresses on bloom versus non-bloom forming phytoplankton species. So the multi-stresses in this aspect would be controlling temperature, micronutrients, carbonate chemistry. In this instance, we're only able to do pH and alkalinity, but we'd be able to calculate DIC from using the relevant equations. And we'd be able to control the pH at 3 settings, where we're looking at the temperature, the pH as it is now, versus what it could be when things are a little not going so great, versus the severe scenario that everyone would go for, where this is when we've gotten to the stage where it is really a problem. So to just get an idea of how our phytoplankton species, and in relation to that, the biological pump would respond to that. And we'd also have to factor in the fact that we are using species that are cultured from this region. So there have been papers where they've spoken about the fact that they believe that the reason why our phytoplankton species live quite happily here is because they have an affinity for low concentrations of nitrate. So they're able to utilize it no matter how low it is, since the variability of nitrate is through, let's say through an annual series, always fluctuates, but they're still able to utilize it. So we'll be able to sort of have an idea also of which of the species has a greater affinity for nitrate with respect to that. And then we'll also be able to actually see if nitrate is the limiting factor, or if these other scenarios actually change aspects in such a way that they're able to utilize another component. And then this would also be of interest for the non-bloom forming species, because we still don't have a consistent agreement of how non-bloom Hab species actually will be affected by high CO2 concentrations. So again, in order to test Malagraf's postulate, we would basically use bloom conditions in order to see if this would actually relate, would advantage one species over the other. And then one of the other aspects also, which we'd actually look at, is that if you look at the CO2, the reason why there isn't much consensus between how phytoplankton utilize the carbon dioxide is that they basically use slightly different systems. So like the dinoflagellates have a low affinity for CO2, whereas the diatoms have a CO2 concentrating mechanism where they have 4 transports. I don't know what I call them. Transport enzymes doesn't sound like the right word, but they have a mechanism in which they can actually utilize the DIC in the water column and able to get it for them. But again, these mechanisms are very species specific. Yeah, so of course, the community that you use will actually affect how your DIC is taken up. So this would enable us to sort of have an idea of how the biological pump would actually react in those aspects. And the reason why those aspects, the reason why I was talking about the fact that they have two different mechanisms of which they take up CO2 is because you also get instances like this, where even though the Benguela is generally a diatom dominated system, you get some instances such as this, where this was during the end of March, during a downwelling season, I showed you from before, and it specifically focused on, there were no dinoflagellates in here. So you have to have some way of assessing how well they utilize the DIC in relation to whether it's diatom or dinoflagellate dominant. And then for the second experiment, here we're going to be looking at how phycotoxin production is actually affected by climate change multi-stressors. And in this instance, the multi-stressors we're talking about is temperature and ocean acidification, which would equal pH. So we're going to start with the Alexandrian tamarindic culture, which is a culture I've had for the longest period of time. And we're going to be assessing the effects of temperature and pH with respect to their toxin production and, of course, the cell growth as well. Oh yeah, and as well, we'll also be looking at how it affects, because the PSP toxins, you get about, saxitoxin is a parent molecule. Actually, that's on the next slide. Okay, I'll explain the profile on the next slide. And then as another aspect to add on to that, once we have an idea of how the toxin production is affected by these changes in temperature and pH, then we'd also go that a step further and see how this affects how mussels and oysters actually assimilate these toxins during that period of time, as in does their feeding rate increase or decrease accordingly. And the reason why for the PSP toxins is so important is because this is a saxitoxin parent molecule, and then it has different analogs. Now, the analog that it has determines how toxic it is in relation to the saxitoxin molecule. So you'll find in some instances that the toxins are sensitive to temperature, changes in pH. So you could actually have it metabolize if you cook the shellfish first, or metabolize in your stomach, and then you'd have different toxicity values. So that would be of interest to the, what are they called? The shellfish industry, because then they could actually use this information to determine whether or not they would actually harvest. Because in a lot of areas that have more information, they're able to actually have trigger levels and assessments and all of that. And it's based on this type of work. Yeah, so the important part here also is that there's limited work out there that actually looks at the relationship between the growth of HABs, how they produce their toxins, how much toxin per cell is actually produced. So this would give us an idea of that relationship linked to an area that is nitrate deficient. And of course, we could use other samples with respect to this, like Gonialis spinifera, as well as some of the pseudonaceous species that are toxic. Okay, for the third and final experiment, this one is sort of more mesocosm-like because of the scale. So we're essentially working with a PhD student from Northwestern University, Monterey. And what his work is trying to do is to understand what the role of dust is with respect to bloom formation. Because if you look at this area, especially where we are, I should have taken a picture this morning. We have hellishly hot temperatures at the moment. we've had wind blowing, we've had dust inclusion. Now during other periods of time, you'd get a bloom within the day or so later, whereas now there isn't much. But I have my ideas of why that isn't, but again, I'll discuss that later. But for this specific aspect is that they took dust from certain riverbeds. He sieved that down so you could have like, I think it was like 0.2 microns in size, anything smaller than that. And we essentially introduced it into these, because these are essentially tanks that have filtered seawater. And then we took a mix of non-organic cultures that we had growing here into all the different tanks. We tested the temperature because we couldn't control it during this period of time. And we took samples for all these other parameters. And the most important thing here would be the dissolved iron. So we want to see, is it the dissolved iron or is it an increase in the macronutrients that are in the water column, or it could be one of the other trace metals that actually causes this positive reaction where you get bloom formation with respect to that. And for the next phase of the experiment, because we have pH regulators, we'll be able to set the pH at certain ranges to be able to add another aspect of ocean acidification, because that could also probably potentially affect the dissolution of the dissolved iron as well as the trace nutrients. And yeah, so the diversity with which we use with respect to the cultures is going to be dependent on what I'm able to grow in the water. But what we tried to do from the previous experiment was to get a good combination of, the chain forming ones, which is, this would be Thalassiocera. Then the ones that are technically, yes, chain forming, but then there's a limited number of cells that are attached to that. This would be Rhizolinea, the single cells, which would be, what is this one? Anyways, and of course, dinoflagellates as well, just to get an idea of if there's an aspect of size variation and if there's an aspect of whether they're chain forming versus non-chain forming and bloom versus non-bloom forming species. And then this would be linked to the input of dust. So I'm going to end off with that, as in this was day 4 of our experiment, where both of these had inoculation and the same inoculant of cultures, because we had a mixture of different species. We added dust to one, no dust to the other, then you actually see the growth. So the point of this now is basically just to put essential data to these aspects in order to get a good relationship that will be focused on the northern Benguela aspects. And with that, I am done.
Is there anybody who would like to ask any questions for Chibo?
I enjoyed the presentation. It was really cool.
Hi, Christo. Thank you.
So these experiments, are they all ongoing at the moment? Do you have some results that you can refer to already in terms of different stresses that you applied? The dust was particularly interesting. Well,
yeah, for the dust one, unfortunately, Monray took those samples. So he's basically working through those results. So the only data that we literally have is that I can show you like growth curves of, what grew better than others sort of thing. But we were trying to sort of, instead of getting lost in the weeds of all the different species, we wanted to sort of that with respect to the dissolved iron. And if I speak out of correction, but I don't think he has that data yet. Yeah. I could tell you once he gets that, because I think that would be really interesting, because it's something that you can visually see from this area also as it is. But we just don't know, whether it's nutrients or whether it's trace metals that cause the bloom formation.
Anybody else would like to ask Chibo a question?
You probably meant besides Kurt, right?
Kurt, don't ask me about Robisco. I don't know.
Chibo,
I'm waiting for you, Kurt.
I must first of all congratulate you for the courage to pick up all these topics, right? That's a lot of courage that you need.
Never again.
But well, other people have made life careers with one of the ones that you are choosing here. Dust
Okay.
experiments are great, and especially the way you are doing them in a kind of a semi-laboratory assay. But do you know what that dust is? You know, if it's just sand from the Omaroro riverbed, then you probably have no idea what you're feeding. Or is anybody analyzing the dust components that are dissolvable in your setting?
The samples that they used, it's based on a paper because he's actually using old samples from. They took samples along the, the riverbeds along Namibia. So that information is there actually. So the idea behind it was to just sieve it out so that we actually focus on the bits that can actually dissolve or actually, release the trace metals. So yes, it is there. The only data that's in the process of being done is the trace metals that actually dissolved in the water. Yeah.
But then you have lots of trace metals and you don't know which one is then the regulating stressor in your experiment, right? Why not just
True.
feed the one you're interested in?
But how do you pick one? The idea of it was we're trying to simulate like a field experiment where it's like whatever's there, and then you just feed them with dust and we see what happens.
Yeah, that's
because not a lot of this is, because Kurt, if you look at it, there's not a lot of experimentation like that done here. There are a lot of areas that have a lot of these aspects that are, categorized like readily, sort of. So I get what you're saying, but this is a phase 1 for us. So phase 2 is going to be in July. Then we can probably say, fine, from the previous data, this was what it actually was in here. So let's rather focus on this aspect. Because the dust has to be in it.
May I have another question?
You can have as many questions as
yes, you can have a question, but it's fine.
Well, this is more a comment. The ribulose bisphosphate carboxylase that you are interested in for the CO2 uptake, right? That's I would say, forget that.
Yeah, no. It's just to mention that there are 2 mechanisms, so that when we look at them with respect to them changing the DIC rates, then we can maybe link it to a specific species. Because what I want to sort of do is tie that link that we have where sometimes you have just diatoms and sometimes just dinoflagellates. So sort of be able to look at the rates of that. But no, I'm not going anywhere near enzymes and those kind of things. I don't have the capability.
Yeah, exactly. That's a very hot topic and it's being studied worldwide. But I would like to go back to what you said at the beginning. With what you're using the word multi-stressors for as an alternative word for drivers or for environmental conditions, right? Just combine them at the same time as it happens in reality. Is that correct? Is that how you use the word multi-stressor?
Yeah, it's a multi-stressor / driver.
Right. And then it dawned on me, we always talked very separately about nutrients in the Benguela, and now you combine that with is it a sink or is it the source, or is it nothing? Because the pump remineralizes the organic matter so quickly, at least the surface pump. And if we put any carbon into the sedimentary sink, then we also move nutrients away. So it's very hard to only use one component as a stressor, like more CO2 doesn't help. If we go along 23 south, and that's a standard line, what does that really tell us about whether it's a sink or a source? 23 south is in a region where you have extremely much terrestrial anthropogenic input from the Walvis Bay area. The southern Benguela is lower in nutrient. The northern Benguela is higher in nutrient because of the direction of the Benguela current. So it's very hard to make any conclusion about that particular question from data along 23S. You're not nutrient limited there.
I know. I'm just trying to. The idea of the first experiment even though, yes, we're trying to mimic what is in the field, essentially takes away that remineralization aspect. You put, 10 species in there, how well they use the nutrients until they get to a point where there are not enough nutrients, and then they basically die off. So from that perspective, you could probably figure out which ones are the nitrate affinity, the ones that have an affinity for nitrate. But then from the field experiment, yeah,
Yeah, see, in my opinion, that's the failure of our very fractionated studying ecosystems. Right?
Yeah, but then
failed, but we cannot combine them.
Okay, but then this would be phase one of the combination. Because then a second phase would be to, let's say, add zooplankton. Then the third phase would be to get, get it to sort of, it's such a stage where you have the microbial community like breaking it down. But where I am now, that is a little too complex, because distinguishing between remineralized nutrients versus what was there before is, yeah, I don't know. It would be difficult.
Yeah.
So I get what you're saying, yes. Yeah. But I still feel like there is still promise in doing the first phase of it.
Oh yeah, absolutely. I agree. Now I would like to ask Nancy. You made a comment in the chat. Would you like to say anything about that?
Okay, I've just remembered that there was a presentation by some lady during our summer school. She presented about the macronutrients. And then one of the things that she indicated was that they realized that now iron was limiting in one of the factors limiting primary production. So I was just wondering if that could be the basis of the use of dust in this study that has been done by Dr. Chibo.
Oh, I think you're talking about, I think it was Sandy. I think Sandy was there when you were there. That experiment was linked specifically to the Southern Ocean because it's a high nutrient, low productivity area. So it took them from all of, the testing, whatever, they realized that the reason why, even though the nutrients are there, the phytoplankton just can't get going is because iron is the limiting factor. But for our system here, iron is not our limiting factor. That's why we're trying to sort of, it's actually quite true in what you're saying, in the sense that we're saying, look, when we have an input of dust into the ocean, you can see that you get a lot of productivity stemming from that. But we're in an area that's technically not iron deficient. So what else does the dust have that it's incorporating into the water column that the phytoplankton need to get going? You can sort of look at it as that.
I know that even if iron is introduced in the form of system, that it has to, and that component I can't remember, it has to be in another form before phytoplankton can take that up. So, and that's the process that you have to maybe check, if I can call it that.
Yeah, it's one of the ions. It's the 3+ or the 2+. I can't remember. Yeah. Something.
So what might be that change? What changes that? And with other studies, just to come back quickly to your trace elements, and what Kurt also said that the Orani papers. There's 2 Orani papers in terms of trace elements all along the Namibian coast. Maybe to look at what is the main ion or that ion's trace elements, and then maybe work from that point. That's just a suggestion. But I think that would be a good afterwards, because with a lot of these ions, you can find them in solution, so we can probably just dissolve it. But then first would be to just say, look, dust input, see what happens, and then teeth together those aspects. So yeah.
Okay. Yes, it's true. The iron 3 oxyhydroxides are less soluble than the iron 2. Right? Yeah. In an oxic environment, that iron 2 immediately oxidizes to the iron oxide or hydroxide. And Nancy, yes, we also had a presentation about 2 weeks ago by Luke Bridgestock. And if you it's online. You can listen to that again. And he also reiterates the importance of iron in the Benguela, because that's what he studied. So iron is still assumed to be a regulating component. Probably not, in my opinion, probably not in the Benguela, because other things have a much quicker turnover there.
Yeah.
Okay.